Hello, my name is Alan Dunton. I'm a non-executive director at Recce Pharmaceutical, and I've had the pleasure of working with the team at Recce for a little over 2 years now. I joined as a consultant early on and later became a director. My background is in medicine. I am a physician in adult medicine, but my specialty was in clinical pharmacology, which is the action of drugs in humans. And as such, I pursued a career in drug development, which was very satisfying, a 40-year career. And now it's a pleasure to be helping a young company just like Recce. I used to be the president and managing director of Janssen Research for Johnson and Johnson. You probably have heard of them. And before becoming the president of Janssen, I was the global vice president of R&D for the R.W. Johnson Pharmaceutical Research Institute, which was another arm of drug development at J&J. What I'd like to do this morning is take you through drug development as seen through the eyes of Recce. I've followed this prescription many times, and it's been successful very frequently. As you know, we're developing an antibiotic. It's called Recce 327. And the way an antibiotic is developed is screening occurs not in animal models, but in cellular systems. And you can actually evaluate whether your drug is efficacious in killing various bacteria or a virus or fungi or cat's-heel species. And then you apply them to animal models to check on the data when you see a strong signal. So we saw a very strong signal against the E. coli bacteria, which is the offending agent for most forms of infections in the urinary tract. And if I can take you to the next slide, we did this study in rats where we infected them with E. coli actually in through the bladder, and the bladder became infected, as did the kidneys and the ureters. And what this slide is showing on the left, when it says efficacy in bladders, it's actually the amount of bacteria that's present in that bladder tissue. So the bladders are removed after the rats are dosed at a certain period of time, beyond 24 hours. And you can see the first gray bar is the vehicle control. They were infected animals. They had a placebo, so they really didn't get treatment. And in the blue bars was a lower dose of Recce 327, 50 milligrams per kilogram. And you can see a rather significant, greater than 1 log reduction in the amount of bacteria present in that bladder tissue. And when animals had been dosed with 500 milligrams per kilogram over that 24-hour period, the decrease was even more dramatic, down to 6.69 logs was present, so greater than a 2.5 log reduction in the amount of bacteria that were in the bladder. And then we did the same evaluation in the kidneys. We ground up the kidneys and looked at how much bacteria were present. And what you can see there at the higher dose, the 500 milligrams per kilogram, there was over a 1 log reduction actually in the kidney. This is really excellent results in this model, which I've used before. So this rang the bell, obviously, that perhaps we have a chance to be an effective antibacterial agent with this product. But obviously, what you have to do then is to go into clinical trials, which we did this year. We went into the phase 1 trials, which look at the safety of your product, as well as the pharmacokinetics, which means how much drug is present in the bodily tissue or sample of fluid that you're taking. So we looked at plasma levels in the blood as well as in the kidney. I'll have to say that the drug was very well tolerated, but what we found, which was very, very pleasant for us to see, was that the primary route of elimination of 327, meaning how does it get out of the body, appears to be through the kidney, into the ureters, and down into the bladder before it's excreted. So if you look at the bottom of this slide, it shows you the concentrations on the left-hand side in human plasma of 327, and on the right-hand side in human urine. And these are comparisons within the same patient, obviously. And as you'll note, in the plasma concentration side, as we went up in dose from 50 to 6,000 milligrams given intravenously, we saw a corresponding, and I'll call it a dose correlated, meaning it went up as much as the dose went up in the plasma concentration of R327. Now, these plasma concentrations are more than enough to be treating bacteria, to effect a kill of bacteria, or in fact, potentially even virus in the human condition or in the human system. But really was a bit of a surprise to us because we hadn't seen this type of thing before, since we hadn't done human trials, is those concentrations in human urine. And to the right, you can actually see the ratio of the urine concentration to plasma. I'll call it on average about a 15-fold higher concentration in the urine as compared to the plasma. So if you're thinking about developing this drug for urinary tract infection, you're in the right place, because the drug is apparently cleared through the urinary tract. Now, we suspect it's being distributed throughout the body, but it clearly seems to concentrate in the urine. So this is a real opportunity for therapy, not only of UTI, but from the plasma concentrations. Could be used as a broader anti-infective treatment for other conditions, and we'll be testing those as we go forward, in addition to urinary tract infections. The next slide is the same single-dose study, which shows the concentrations in the urine, and you can see the dramatic increase as we go up in dose. In the slide on the left, the purple bars are the actual concentrations in human urine, which is, let's call it, its maximum is about 2,200 micrograms per mil, and this is a very high level. Now, what's very important, and I asked myself the question, yes, but was it tolerated in the bladder and in the kidneys? It was very well tolerated. There was no sign of any irritation or any other issue with the urine that we noted. There were no red cells in the urine, no blood, absolutely anything like that. Patients did fine with this drug. So it's not only well tolerated in the body for all the other things we looked at, but in the urine as well, in the bladder and through the urinary tract. So this is a very compelling profile for an urinary tract infection drug candidate. I've developed several products for urinary tract infection, had those indications or that indication, and this is a very exciting result. And again, here are the plasma concentrations, and as you can see, a very nice rise as you go up in dose of the human plasma concentration. And again, as I noted earlier, these are reaching plasma concentrations which can be efficacious to engage bacteria, not only in the human vasculature, but also in other parts of the body, as we are suspecting that it's broadly distributed throughout the body. So in summary, then, we've completed preclinical studies in UTI model in the rats, which, as they say, follow the data, and that's what we did. We saw a signal in the rats. And then again, in the phase 1 clinical trial, which we're completing, given as a single dose, R327, the drug was safe, well tolerated, and we saw very high concentrations in the urine. So another very positive signal for not only treating urinary tract infections, but as we know from the data that the E. coli is the most common bacteria which causes sepsis in humans coming in through the urinary tract system. So we have very strong data in E. coli and other bacteria. We have high concentrations in the urine, so we'll be doing phase 2 trials as well. The thing that's very important here and that's critical is we feel we have a chance to have a single dose cure using R327 with these high concentrations, plus the mechanism of action, as you've heard by other speakers, mentioned by other speakers. We have a real shot here of a single dose cure. There's a very high percent of, especially in women, who get chronic urinary tract infections. They're put on many different antibiotics, which eventually all they do is make the E. coli resistant to the new antibiotic they're being put on. Given our mechanism, we feel this is not going to happen with Recce 327. So we'll be doing single and multiple dose evaluations of the urinary tract infection, different types of UTI. And then with a positive signal there, we can move into what our ultimate goal is, really, is to have this drug in sepsis in patients who are very sick. So they'll be in an ICU type situation, and they'll be evaluated for the efficacy of 327 in urosepsis. So there's a very exciting period of time for the company moving forward. And, this is one of the best drugs I've seen in terms of a urinary tract infection candidate going forward. So, I've had fun in this career that I've had in drug development, and it's been a pleasure working with Recce, and especially with Recce 327. So I thank you for your attention, and have a good one. Take care.